Okay, thank you. Let's get started. Good evening. So my name is Gopal Babu. I'm an associate professor in the Department of Cell Biology and Molecular Medicine. And today's lecture, we're going to cover the basic aspects of cardiac muscle. Um, I mean, I'm going to give you only the basics, not the cardiac physiology. We're going to see some basic anatomy and then a little bit on molecular biology of muscle contraction and some examples with disease models. Um, and so if you want to, read more about cardiac tissue, you can see the additional reading materials which are available in Physiology of the Heart or Heart Physiology. You can see some of them, not all of them, but some of them. But for exam point of view, the class notes are sufficient. You don't need to go for additional reading. Okay. Okay. So it's not moving. Uh, you know, yeah, I tried. Okay. Okay, so the lecture is organized in such a way, so you're going to see the basic anatomy of cardiac muscle, and then we're going to see what triggers cardiac contraction and the role of calcium in muscle contraction, and a little bit on metabolism, signal transduction. And the last two parts, we're going to see the molecular basis of heart disease with an example of cardiac ischemia. So we're going to start with the anatomy, basic anatomy and structure, which are very important to understand the cardiac muscle function. So as you know, like the heart has the two sides, the right side and left side. And each side it has atrial chamber and a ventricular chamber. And the left side and right side and the atria and ventricle, they do not, the left and right side of the atria and left side and right side of the ventricle, they do not communicate directly, except in some congenital anomalies where the heart diseases, you can see some kind of like hole between the atrial septum or ventricular septum, so they can kind of miscommunicate and cause the cardiac pathology. And each side, as I said, it's made of an atrium and a ventricle, and they are separated by valves. And so I'm going to give you the names of certain organelles, the valves, which are very important with respect to cardiac pathology. So you should remember all these names, for the exam point of view. And it's also very easy to remember these things. So the heart cardiac muscle is, always connected with blood vessels. The left atria or right atria or ventricles, left or right ventricles, they are all like connected with blood vessels. And to remember the names, the atrium is always receives blood from veins, or in other words, the atria is connected to veins. In the case of right side, you can see the right atrium is connected with the vena cava, which is superior and inferior vena cava, which collects the blood from all parts of the body and takes into the right atrium. And in the case of the right side, the left side of the left side atria is connected with pulmonary veins. So which connects the pulmonary veins with the pulmonary system, with the lungs for receiving the blood from the lungs to the left atrium. And the ventricles, whatever the blood vessels is connected to the ventricle is called as arteries. So in the case of right side, it is a pulmonary artery, and in the left side, it is called aorta. So aorta is the major blood vessel which supplies blood to the all parts of the body, or it is involved in systemic circulation. And the blood flow is unidirectional. So the valves are very important, and it plays an important role in preventing the reverse flow of the blood from one chamber to another chamber. And the names of the valves are like, based on the structural arrangement of the valve. For example, the valve which separates the right atrium from the right ventricle is called tricuspid valve. If you look at the valve, you will see a three cusp-like structures. So that's why it's called tricuspid valve. And the valve which separates the right ventricle from the pulmonary artery is called pulmonary valve. And the valve which separates the left atrium from the left ventricle is called mitral valve or bicuspid valve. It has a two cusp-like structures. And the valve which separates the left ventricle from the ascending aorta is called aortic valve. So both the aortic valve and pulmonary valve, also called as semilunar valves. So if you look at the structure of the aortic valve and pulmonary valves, they look like a semilunar shape. So that's why it's collectively called as semilunar valves. So these valves are very important in preventing the backflow of the blood. And if there is any defect in the valves, we are going to see that that will lead to the cardiac pathology. And what is important here is the arrangement of valves, how these valves are arranged. So if you take a cross section at the junction of atria and ventricle, and you will see all the valves are arranged in a particular orientation or in a plan. So you can see this is a idealized image, and you can see all the valves are located in one position, and they are placed in a specific structure called as central fibrous body. So the central fibrous body is kind of an insulator. So it prevents the direct connection between atria and ventricle during electrical stimulation. So if you look at the cardiac contraction, the contraction starts at the right atria from the SA node or sinoatrial node, and then it propagates to the left atria. Then it will be propagated to the ventricles. So, and then if you look at the contraction, the atria contracts first and followed by the ventricular contraction. So there is a delay between atrial contraction and ventricular contraction, and that delay is important for the proper, receiving of the blood from blood vessels to the atria and from the atria to the ventricles. And this, the prevention of the passing of the signals from atria to the ventricle is prevented by the central fibrous body. However, in the central fibrous body, you will see a place called AV bundle or His bundle or atrioventricular bundle. This is a fibrous, the nervous ending. That's where the signals from the SA node will be transmitted to the ventricles. So once the signal comes to the AV bundle or His bundle, then it will propagate into the ventricle and stimulates the ventricular contraction. So, this central fibrous body therefore plays an important role in maintaining the proper atrial and ventricular contraction. And then if you look at the anatomy of the heart, although the muscle, the cardiac muscle, looks like a thick muscle bundle, it's made of several parts depending on what type of cells present and how it contributes to cardiac contraction. So the major portion, if you take the cross section, the major portion of the cardiac muscle, which is involved in muscle contraction, is called as myocardium. So if wherever you see myocardium, that indicates, sometimes people say myocardium for the whole heart, but the myocardium indicates the working part of the cardiac muscle, so which is made of mostly cardiac myocytes. And then the second layer is the endocardium. Endocardium is the layer which is present inside the heart, which has a direct connection with the blood. It's not directly connecting with, it's not directly, connecting with the blood, but it is also, separated by a layer called endothelial layer. And this endothelial layer separates the endocardium or the cardiac muscle from direct communication with the blood. And then the outer layer of the heart is called as epicardium. The epicardium also forms the visceral layer of the pericardium. And then the outer layer of, another layer is called parietal layer of pericardium. These two layers kind of form a small, structural separation between the cardiac muscle. And these pericardium also contains a cavity called pericardial cavity, which is filled with a fluid called pericardial fluid. So the importance of pericardium is it is acting as a protective seat surrounding the myocardium. So it kind of lubricates the outer heart wall. And also it acts as a shock absorber because the pericardial fluid, which is present in between the pericardial layers, it kind of act as a shock absorber. And also the pericardium is attached to other parts of the body, to the sternum, and also to the other parts of the body. So it acts as it kind of holds the heart in place. And since it has a double layer and a fluid, it also acts as a barrier for infections. If anything wants to go through, it has to go through the pericardial cavity, so it acts as a barrier. And also it helps prevent the heart overexpansion. So during contraction and relaxation, the heart can expand, and this expansion can be, regulated by pericardium. So the pericardium has, all these kind of functions. The other important parts of the heart are coronary arteries. The coronary arteries are the arteries which supply blood to the myocardium. So we're going to see that a little bit in detail. And then the capillaries are the microvessels, which kind of connects between the cardiac myocytes to supply oxygen. And why this anatomy of the heart is important? Because many of the heart diseases are associated with the cardiac structures. The next slide, you're going to see the importance of various cardiac structures. So any kind of heart disease which is associated with a dysfunctional myocardium is called as cardiomyopathy. So the cardiomyopathy is usually associated with dysfunctional myocardium. And it could be ischemic, heart disease, or hypertensive associated heart disease, or congenital or valvular, disease. It's all, it's going to cause the dysfunctional myocardium, and that's called cardiomyopathy. And if the cardiomyopathy is not treated, and then it's going to be heart failure, which is the insufficient cardiac function. And the endocardium is also associated with a disease called endocarditis, which is nothing but the inflammation of the inner layer of the heart. And this could be caused by the bacterial infection or fungi. Whatever comes in the blood can, infect or cause the inflammation of the endocardia, or the endothelial cells in the endocardium, and it causes endocarditis. And pericardium also associated with a disease called pericarditis, which is, again, nothing but the inflammation of the pericardium. And it's usually a viral origin, but it could be caused by bacterial or fungal infections. And the pain induced by pericarditis will, kind of mimics the heart attack. So there are other terms. For example, if the pericardium is, as I said, it has two layers, the epicardial layer and outer layer. And in between, you will see a pericardial cavity and pericardial fluid. And this pericardium is fibrous in nature, and it is very rigid. But during some infection, you will see some kind of effusion. So that will result in pericardial effusion, the entry of the more accumulation of fluid inside the pericardial cavity, and that will compress the myocardium, and it's called cardiac tamponade. So if you see pericardial effusion or pericardial tamponade, that's all the impairment of the pericardium, which will affect the ventricular or the myocardial function. And then coronary arteries are associated with ischemia. At the end of the lecture, you're going to see what is ischemia and what can cause the cardiac ischemia. And mostly it is associated with the obstruction of the coronary arteries, which supply blood to the myocardium. And capillaries are involved in angiogenesis, especially if there is an ischemic conditions, meaning that there is no blood supply to the myocardium. And then at that time, the angiogenesis will occur. The capillaries will start to proliferate, and then it will try to kind of provide the oxygen to the myocardial cells. And it depends on the type of stress. It could be pathological or physiological. So we'll see that later on. Okay, the valvular heart disease. So we have seen like four different valves: the aortic valve, mitral valve, tricuspid valve, and pulmonary valve. And all these valves can also be affected. There are two different types of the valvular diseases. One is stenotic disease. The other one is regurgitation. So the stenotic disease is associated with the narrowing of the valve, and the regurgitation is insufficient function. So the function is not proper. So if it occurs in aortic valve, then it is called aortic valve stenosis. And similarly for mitral valve or tricuspid valve, it's called as aortic valve stenosis or mitral valve stenosis. Or similarly for aortic regurgitation or mitral regurgitation. So the most common ones are aortic valve stenosis, and then mitral regurgitation. These two are the common ones, but you'll also see a lot of patients with pulmonary valve stenosis or regurgitation, which can cause the cardiac defect. So the next two slides, the next slide is going to differentiate what is stenosis and what is regurgitation, and how and what causes the stenosis and regurgitation. So the aortic stenosis, as I said, this is associated with the opening of the valve. So if the valve does not open, in aortic stenosis patients, and the common causes are congenital abnormal. So during development, there may be a defect, and the valve cannot open properly because of that, or it could be caused by some infections, including the rheumatic fever is one of the major causes of the aortic stenosis. And in aged patients, you will see the calcification of the valves, which is shown here. So this is the normal, opening of the aortic valve. And, in case of stenosis, it could be of any origin, and you will see the improper opening of the aortic valve. In the case of congenital anomalies, you will see only the two, the valves, not three. So even, that's very less common compared to the aging-associated stenosis. And regurgitation is exactly the opposite. Here, the valve opening does not close completely. So, and because of the valves are not closed properly, the blood can leak backward into the heart. So, in the case of aortic stenosis, if it is not closed properly, and the blood will be coming back to the ventricles during contractile cycle. So you will end up with volume overload in the ventricle, and then that can cause the hypertrophy and heart failure. And aortic regurgitation is commonly caused by high blood pressure, bacterial infection, and untreated syphilis or injury. Or again, the aging can also affect the valve function. So the aortic stenosis and aortic regurgitation are also common cause of cardiac dysfunction in aged patients, or it's associated mostly with the aging. So the same mechanism is also applied to the other valves like mitral regurgitation or mitral stenosis. Those are also very common, especially with these two are associated with aging. Okay, so the aorta is the major blood vessels which supply blood to all the organs. It's involved in systemic circulation, and the first branch of the aorta is called as coronary artery. So the coronary arteries are the arteries which provide blood supplied to the cardiac myocyte muscle itself. And there are two different coronary arteries, the left main coronary artery and right main coronary artery. It goes in left side and right side respectively. And the left main coronary artery is very small in length, and then it immediately divides into left anterior descending, called as LAD, also called as anterior interventricular artery, and which travels along the interventricular groove. And then the other branch is called circumflex artery, so which travels the posteriorly along the groove between the left atrium and ventricle. So basically, the left main coronary artery, supply blood to the left part of the myocardium. The right main coronary artery, it travels between the right atrium and the ventricle and takes care of the right part of the heart. Similar to the left main coronary artery, the right main coronary artery is converted to posterior descending artery. It's called as PDA. And in 80% of the humans, the posterior descending artery is derived from the right main coronary artery. And in 20% of the human, the posterior descending artery is derived from circumflex artery. So it has no physiological relevance, but it has a pathological relevance because if there is occlusion in the left main coronary arteries, in case of the humans with the posterior and descending artery derived from circumflex artery, it's going to affect the whole heart. But if it is derived from the right main coronary artery, it is going to affect only the left part of the heart. So it has a clinical relevance. You don't need to remember all those things. Mainly the posterior descending artery is involved in supplying blood to the right part of the myocardium. And we're going to study what is atherosclerosis and how it is going to affect the coronary artery in one of the area of the lecture. So the coronary sinus is the veins which receives the blood from the myocardium. It's mainly from different cardiac veins called small, middle, great, and oblique cardiac veins. And everything will be open into the coronary sinus, and then it will go to the right part of the circulation for purification. So what you have to remember is the left main coronary artery and right main coronary artery, and then how the LAD and circumflex arteries are derived, and posterior descending artery is derived from right main coronary artery. Okay, so this is the basic structure of the myocardium and associated blood vessels and valves. So why it is important is many of the diseases are associated with the valves or the structure of the myocardium. Okay, so the pump function of the heart, it's very simple. It has two different circuits, the pulmonary circuit and the systemic circuit. So, the oxygen, poor oxygen and more carbon dioxide rich blood is collected on the right part of the myocardium, and then it will be pumped to the circulatory system, pulmonary circuit for purification. And then once oxygenated, it will enter the left part of the myocardium, and it goes into the main aorta, and then it goes to the systemic circulation. So what is important here is the pump function. So the cardiac muscle has to pump the blood, from the ventricular chamber to the arteries to supply blood to the entire body, as well as for the purification to the lung system. And this has a pathological importance, but under normal physiology, this is how it works. But in a pathological conditions, during heart failure, you will see backward failure or forward failure depending on the circulation. So for example, if it is a pulmonary circuit defect, then it is called backward failure. And if it is a systemic circuit failure, then it's called forward failure. Okay, so the cardiac muscle is the major portion of the myocardium which is involved in cardiac contraction. And then if you take the myocardial section, and if you look at by H&E staining or by immunostaining, you can see with the H&E staining, you can see all the red areas are representing the cardiac myocytes. But you will also see a lot of the voided areas, which is not stained by H&E. H&E is hematoxylin and eosin, which stains specifically the muscle cells. And you will see a lot of areas which are not stained by H&E. So that suggests that the cardiac muscle contains, a lot of other cell types other than myocytes. And then in the immunostaining, people have shown that, the red one indicates the myocytes, which is stained by anti-myomycin, which is specific for myocytes. And then the blue is actually, which marks the fibroblasts, which is for anti-vimentin. And you will also see some green, which is a connection 43, and TAPI is in yellow-green patches. So what it indicates is like, if you take a section of the heart, and then if you look for the myocytes and non-muscle cells, you will see large number of myocytes. I mean, large area of myocytes, not numbers. But if you count the number of cells, different type of cells, the cardiac muscle contain only 30% of myocyte, and 70% of them are non-myocytes, which includes fibroblasts, endothelial cells, and smooth muscle cells. However, if you look at the area, the 30% of the myocytes occupy the most of the cardiac muscle. So that suggests that the myocytes are the major functional unit of the cardiac muscle, which is involved in contraction and relaxation. And the other cell types like fibroblasts, endothelial cells, and smooth muscle cells, they act as a supportive system for the cardiac muscles. Okay, if you look at the cardiac myocytes, like any other cell types, you will see a plasma membrane, which is called a sarcolemma. And the important feature of the cardiac sarcolemma is like you will also see an extension of the plasma membrane, which goes deep inside the myocyte, and which is called as T-tubule. So the T-tubules are nothing but the extension of the sarcolemma or plasma membrane. And this structure is very important for the proper contraction and relaxation of the cardiac muscle. And in addition to the sarcolemma, you will also see a deep, the network of fibers, which is called as sarcoplasmic reticulum. Sarcoplasmic reticulum is nothing but the modified form of endoplasmic reticulum. So in other cell types, you will see endoplasmic reticulum. There are two types like smooth or rough endoplasmic reticulum, depending on the presence of ribosomes. In the case of cardiac muscles, in addition to the endoplasmic reticulum, you will see a modified form of sarcoendoplasmic reticulum. And this endoplasmic reticulum is the calcium store. And this calcium is important for muscle contraction. So we're going to see that. And apart from these two, and you will also see a large amount of mitochondria because the cardiac muscle is repetitively contracting and relaxing. So it requires large amount of energy in the form of ATP, which will be provided by the mitochondria. So these three are the major components of cardiac myocytes. In addition to that, you will see all other organelle which is present in other cell types. So if you, there's one study in which people have isolated the various components of a cardiac myocyte, individual cell, and they analyzed how much volume it occupies. And if you look at it, the myofibrils are nothing but the contractile proteins, which are involved in muscle contraction like myosin actin, and that occupies almost 50% of the cell volume. And then the mitochondria occupies 36% of the cell, and then the sarcoplasmic reticulum 3.5, and then the remaining is the other part of the cell, like the nuclei, Golgi, or whatever the other cellular components. So this suggests that the cardiac myocyte, contains mainly the contractile proteins, mitochondria, and sarcoplasmic reticulum, which is needed for the calcium store and for muscle contraction. So, and if you look at the cardiac muscle, the cardiac myocytes are connected to each other. So this is different from skeletal muscles. If you studied skeletal muscle, probably you would have known that the skeletal muscles fuse together and form a myotube. So in cardiac muscle cells or cardiac myocytes, they don't fuse together, but they are connected by a specialized membranous structure called intercalated discs. And then each cardiac myocytes they are usually single nucleated. Sometimes you will see two nuclei, but mostly they are single nucleated and with a diameter of 25 micron and the length of 100 micron. And this is a normal size. However, during pathological conditions, you will see the enlarged myocytes, which will be more than 100 microns in length. And the cardiac myocytes are connected by a specialized, membrane structure called intercalated disc. And this intercalated disc contains a lot of communication systems, which kind of brings the two myocytes together functionally. So, and in case of skeletal muscles, since these muscles, the skeletal muscle cells are fused together and form a myotube, it's called syncytium. And this kind of syncytium is absent in cardiac myocyte. However, they are functionally syncytium in the sense, if you take the cardiac muscle, the atria contracts at one time, and then the ventricle contracts another time. So they are functionally atria works together and the ventricle works together. Okay, so the intercalated disc has three types of membrane-to-membrane contact, and they are named here: fascia adherens, desmosomes, and gap junctions. And these three are the major communication method between the cardiac myocytes. So the fascia adherens is nothing but a structure which contains actin filaments. So the fascia adherens contains a large number of different type of proteins, but the major one is the actin filaments. The actin is part of the contractile unit. So if one myocyte contracts, and then it will transmit the force through the actin to the fascia adherens, thereby it will stimulate the adjacent myocyte. So they contract at the same time. And then the second type of communication is through desmosomes. Desmosomes is again a protein complex. I'm not going into the details, but the desmosomes contains mainly desmin. And again, the desmin is also connected with sarcomeres or with the contractile proteins. So if one cardiac myocyte contracts, then it will transmit the force to the adjacent myocytes through the desmosomes. So both fascia adherens and desmosomes, they promote the force from one cell to another cell. In other words, the force will be transmitted from one cell to another cell for the coordinated contraction of the muscle through the fascia adherens and desmosomes. The third one is gap junction. The gap junction is mostly present in the longitudinal portion of the interdigitation. If you take a sections and it will be in the communication between the intercalated disc, not on the sides of the myocytes, and they serve as a low resistant pathway between the cells. You will see it in the next slide. This is different from the other two, the fascia adherens and desmosomes, because it can kind of transfer the electrical current from one cell to another cell, and also the small molecules like potassium ions and sodium ions. Those ions can also be transmitted from one cell to another cell through gap junctions. So the gap junction is made up of connections, and each myocyte has a hemichannel, which is made of six connection proteins together, and then it will come up adjacent. The connections will join together and form a path. So through this channel, the small molecules or the potassium ions, which are involved for the action potential or for the electrical current, which will be transmitted from one cell to another cell. So if you compare all three, the communication methods, the phase adherence, and the desmosomes, they are involved in transmitting the force from one cell to another cell, and gap junction is involved in transmitting the action potential from one cell to another cell. So that's the difference between the all three communication methods, which are located in the intercalated disc. Okay, so and then the contractile apparatus is the major portion of the cardiac myocytes. So almost like 50% of the cardiac muscle is occupied by the contractile proteins. They are arranged in the form of bundle of myofibrils. And each myofibril contains a lot of myofilaments, and each myofilament contains the contractile unit, which is nothing but myosin and actin. So these muscles are called as striated muscles because if you make a section and looking at the muscle in an electron micrograph, you will see a striated pattern. And also you will see a lot of the light and dark areas. And based on this, the light and dark appearance, they were called as thick filament or thin filament. Okay. And so this is the electron micrograph, and each one is the sarcomere, and you can also see mitochondria, and they are named based on the structure. Okay, so the idealized image is shown on the right side, and you can see a Z line, which is shown here. This is the Z line. And the area between the Z line is called as sarcomere. So sarcomere is the area which is located between two Z lines. And these two Z lines contain thick and thin filaments. And the thick filament contains myosin and other supporting proteins like myosin binding protein C, titin. You will see it in the next slide. And thin filament, which contains actin and troponin complex, which are involved in muscle contraction. The naming of the zones, like in the electron micrograph, is based on how they are organized. So you don't need to worry about the names, how it is developed. So the myosin, which is a thick filament, originates from a line called M line. So M line means the myosin originating line. So the myosin originates from M line and goes on both directions of the Z line. Okay, but you can also see the myosin is not attached to the Z line. On the other hand, the actin filament, which is a thin filament protein, is attached directly to the Z line. Okay. And then only the thick filament alone is called as M band, which contains only the myosin. And then this light, you can see it from here, the light area is called as I band, which shows only the thin filament. And then the overlap of thick and thin filament is called A band. Okay, so that's how it is named. Not only for cardiac, also for skeletal muscle. But what you should know is thick filament and thin filament. And thick filament contains myosin, which is not directly attached to the Z line. And thin filament contains actin, troponin, and tropomyosin, and which forms the thin filament and which attached to the Z line directly. Then what attaches the myosin to the Z line? The myosin is attached to the Z line through another protein called as titin. So titin is one of the biggest protein in the cellular system, and it binds the the Z line to M line, or basically it kind of attaches the myosin to the Z line. And the titin is like a coiled structure, so it acts like a spring. So it prevents the overstretching of the sarcomere. So it controls the sarcomeric contraction length. So, titin is therefore mostly like, modulating the stretching of the sarcomeres. And then there are other proteins like tropomodulin. The tropomodulin regulates the thin filament length, which is actin filament, and myosin binding protein C, which attaches the myosin to titin. And there are other proteins like Z-line proteins, which contains actinin, desmin, and capsi protein. So I'm showing this mainly because how these cell-to-cell communication occurs. For example, you can see the Z line contains desmin. Desmin is part of the desmosome in the intercalated disc. So if the contraction occurs in one cell, it will be transmitted to the next cell through the desmin also. And M line also contains the origin of the myosin, which also contain other proteins like myomycin, M line protein, and creatine kinase. And creatine kinase is the important one because which converts the creatine phosphate to ATP to feed the myosin for its contractile purpose. So the sarcomeric structure, therefore, not only contains the contractile apparatus or contractile protein, but also contains other signaling molecules and regulatory molecules, so that will kind of modulate the actomyosin interaction. So in addition to the sarcomeres, you will also see large amount of connective tissues in the cardiac myocyte, which are composed of collagen. So you will see all type of collagen 1 to 10. All collagens are present in the cardiac myocyte at a different level. Some of them are in the basement of the membrane. Some of them links the sarcomeres together. And you can see some of the collagens, they kind of connect the cardiac myocytes together, and also it holds the capillaries in place. So the collagen play, it kind of provide a structural integrity to the cardiac muscle by keeping all the cardiac myocytes together and involved in the coordinated contraction of the cardiac muscle. And the sarcomeres are also connected to the plasma membrane. So, we have seen the cell-to-cell communication through the intercalated disc, but also the sarcomeres connected to the extracellular matrix through the plasma membrane or the sarcolemma. There are several mechanisms by which it communicates with the extracellular matrix. So if whatever happens, if you give any kind of stress to the extracellular matrix, it will be transmitted to the cardiac myocytes, and then the myocytes will contract accordingly. So one of the major the mechanism is through the integrin tallin vinculin system. So this system is not only like connecting the plasma membrane to the sarcomere, but it is also involved in signaling the extracellular signaling to the myocytes. This integrin system is more important during pathological conditions. You will see the activation of the integrin and several signaling molecules which phosphorylate several proteins to modulate the contraction. So the integrin is a transmembrane protein. At one end, it communicates with the extracellular matrix through the collagen, and then the other end, it interacts with the tallin, which is a cytoskeletal protein, and then the tallin interacts with vinculin. Vinculin has globular head and a tail region, and the globular head interacts with tallin, and the tail is interacting with alpha actinin. The alpha actinin then communicate with the actin. So what it suggests is like actin is a contractile protein. So if if there is an extracellular difference in extracellular matrix, and the signals will be transmitted through the integrin, and it goes to the tallin and vinculin system and stimulates the actin molecule, so the contraction will occur. I mean, it's a more complicated system, like depending on various stimuli, the cardiac myocyte will adjust according to its signals, and then it'll contract and relax differently. Okay. And the other important part of the cardiac contraction is the conduction system. So the actual signal or the the action potential of a cardiac muscle is developed in first in the right atria, and then it will be propagated to the left atria, then it will go to the ventricle through the His bundle that I showed you in the first few slides. There is a central fibrous body, so it will be transmitted to the um, His bundle, then it goes to the ventricles. So this system, the entire conduction system, is very important because this is the first step in a cardiac contraction. So once the conduction system is stimulated, then it will be converted to mechanical stimuli, then the cardiac muscle contracts. And then the first step is the signal, which is generated in a specific type of cells called SA nodal cells, which is located in the right atrium. And it is also called as natural pacemaker of the heart. So from this node, the action potential or the current, it starts and then it spreads throughout the right atrium and then to the left atrium. And this SA node generates 70 to 80 impulses per minute, which indicates the heart rate. So if you see heart rate means, that's the action potential which is developed in the SA nodal cells, which is about 70 to 80 per minute. And then this signal will then goes into the AV node or His bundle, which is located in the central fibrous body. And for that, it will take 0.1 second, time delay. And because of the central fibrous body, the signal cannot go into the ventricle directly. And this is very, very important because once the atria contracts and then the blood will be pumped into the ventricle, then the ventricle contracts and then it pumps the blood out of the heart to either to the systemic circulation or to the pulmonary circulation. And so there it needs some kind of delay, and that delay will be provided by the central fibrous body. And once the signal comes to the AV node, and then it goes to the AV bundle, then it branches into both left and right side of the heart, and in each bundle then further divided into an extensive system called Purkinje fibers. So the Purkinje fibers are the final end of the entire the conduction system, which stimulates the individual cardiac myocytes. So the Purkinje fiber cells connect with the ventricular myocytes, which is the final path for the cell-to-cell communication within the heart. So once the SA nodal cells started to generate current, and that current will be then further goes into the ventricle through the AV bundle and then Purkinje fibers, and then the entire heart will then triggered and then undergoes the contraction. Okay, so in the first part, we have seen randomly, some structures, ventricular part, some important valves, and the conduction system. But from now onwards, we're going to see a little bit details on how this contractile system is working and what triggers cardiac contraction and how it will be propagated from atria to the ventricles. Okay, so the contractiles, the muscle contraction or the cardiac contraction occurs in two stages. First, there is a conduction system, which triggers the cardiac muscle, and followed by a mechanical contraction, which pumps the blood in and out. So the first part, which is the conduction system, is called excitation. So the cells are getting excited, and then that excitation will be converted to contraction, the physical contraction. And there are a lot of intermediate steps which will be operated by various ion channels and contractile proteins. And so, and this entire process is therefore called as excitation contraction coupling. So once the action potential or the electrical signal is activated, that will be converted to a physical contraction through several processes, and the whole process is called as excitation contraction coupling. So you're going to see the EC coupling or the excitation contraction coupling for the next few slides. So that indicates the conversion of an electrical signal to a mechanical stimuli. Okay. So what is an electrical signal? So if you take nerve cells and it has some kind of electrical potential, so you can measure the electrical potential using electrode. Same way, the cardiac muscle or cardiac myocyte also has some kind of electrical potential within the cardiac myocytes. And if there is a change in the electrical potential, and that's going to change the contractility of the myocytes. And if you take the individual cardiac myocyte and then if you measure the electrical potential by inserting the microelectrode, you can see some kind of current going on between the cardiac myocyte. And if you measure this, the current in a resting condition, when the myocyte is not in a contractile stage, and it is called as resting membrane potential. So resting membrane potential meaning that the myocytes are not in a contractile stage, they are in a resting stage. So if you measure the electrical current at that stage, and you will see the membrane potential will be minus 85 millivolts. And this kind of current, the minus 85 millivolts, is determined by the concentration of positively and negatively charged ions across the membrane, and also the permeability, relative permeability of the ions through the cell membrane, and finally the ionic pumps that transports the cell membrane. So it's basically the positively and negatively charged ions are responsible for maintaining the resting membrane potential. And what are the ions which maintains the membrane potential? They are, sodium and potassium are the major ions which determines the membrane potential. But chlorine also plays an important role. But under normal condition, mostly sodium and potassium are the major ions which determines the uh, resting membrane potential. So the sodium and potassium in a resting conditions, like you will see at different levels of sodium ions and potassium ions in and out of the cells. And other important factor is in a resting cell, the calcium cannot enter into the cell. So the calcium is impermeable to the myocytes when the cells are in a resting membrane, has a resting membrane potential. So this is very, very important because without calcium, the muscle cell cannot contract. So in a resting condition, if you take the myocyte is rich in potassium and very poor in sodium. It means a very less amount of sodium. And this is exactly opposite for the extracellular matrix. So you will see large amount of sodium and less amount of potassium. So there is an imbalance between the sodium and potassium ions in and out of the cells. So this kind of the imbalance is maintained by a pump called sodium potassium pump. So the sodium potassium pump, what it does is it pumps the sodium and potassium in and then sodium out. So thereby it maintains the high concentration of potassium inside and low concentration of sodium inside. So since it is working against the gradient, it requires an ATP molecule for its function. So the sodium potassium pump itself is an ATPase. So it is also called as sodium potassium ATPase. Sodium potassium pump is also called as sodium potassium ATPase. So it uses the ATPase and then it maintains the high concentration of potassium inside the cell. So during this condition, the plasma membrane is impermeable to calcium. So the calcium channels won't get activated until the resting membrane potential is changed. So the calcium cannot enter into the cardiac myocytes. Okay. So if you look at the concentration of potassium, which is really high, around 150 millimolar inside, and it's around 4 millimolar outside, making a chemical gradient. And it is exactly opposite for the sodium. You will see around 145 millimolar outside and very low inside. And again, as I said, it is maintained by the sodium-potassium pump. So here, the other important thing you should know is the conductivity of the ions. So if you look at the potassium, potassium has a very high conductance compared to sodium ions, meaning potassium can easily leave the cell out. Although the sodium potassium ATPase is pumping a lot of potassium inside the cell, the potassium at the same time can leave the cell immediately. But however, the entry of sodium is very, very slow. And because of this feature, what happened was when the potassium concentration is very high inside, it'll started to leave the cell. At the same time, since the potassium is positively charged, it will attract all negatively charged molecules, including amino acids and everything, and it will leave all the negatively charged molecules in the vicinity of the plasma membrane, and it will leave the cell out. So now you will see a negatively charged membrane inside and positively charged outside. And this is a resting membrane potential. Okay, so in a resting membrane potential, you will see negatively charged, inside and positively charged outside. So once this entire system, the resting membrane potential, is disturbed, then the sodium will come in quickly, and then by activation of some sodium channels, and then it will change the membrane potential. So you will see the depolarization of the membrane, so which is shown in the next slide. Okay, so once the action potential develops, the electrical stimuli occurs from the SA node to the AV node to the cardiac myocytes. The cardiac myocyte will undergo membrane depolarization. So this membrane depolarization will activate the calcium channel. So then the calcium will enter into the cell, then it will stimulate the muscle contraction. So in a resting membrane potential, the calcium channels are inactivated. And then once the action potential develops, there will be a membrane depolarization and activation of calcium channel. Then the calcium channel will activate internal calcium store. Then the muscle contraction occurs. Okay, so what is action potential? Action potential is defined as the sequence of depolarization and repolarization. So whenever you see action potential, that means the membrane is undergoing depolarization and repolarization, which occurs something similar here. So in the normal conditions, you will see the negative charge inside and the positive charge outside. Once the membrane depolarization occurs, and there will be a shift in the charges from negative to positive, and this results in the action potential development or the membrane repolarization. And this action potential is controlled by the influx and efflux of sodium and potassium ions specifically. And once the action potential is transmitted, action potential is developed in one cell, it will be transmitted from one cell to another cell by a domino effect. So every cell will then get activated. So that way, the entire cardiac muscle contracts at the same time. So this kind of the membrane provocation or the action potential provocation is important for the coordinated contraction of the cardiac muscle to pump the blood. Okay, if we go further detail of action potential, how the action potential develops and what are the channels involved, and this slide basically explains what is the different steps in action potential. So the action potential has 5 different phases, and these 5 phases are depending on the activation and inactivation of various calcium channel and potassium channels and sodium channels. So in a working cardiac myocytes, if you measure the action potential, and it will be like this. So you will see a peak and then a small dip and followed by a repolarization step. So this peak is a depolarization and then followed by repolarization. So this is divided into 5 different steps based on the activation of various channels. So the phase 0 is the influx of sodium. So as I said before, the sodium ions are very, very low inside the cell in a resting membrane, in a resting cell. So once the action potential develops, the sodium channel get activated. That induces the membrane depolarization. So the sodium, you will see a peak, and then there will be a activation of the potassium channel called I2 channel, which is a efflux of potassium. This one will limit the entry of sodium, and thereby it will prepare the cell for the repolarization step. And then the phase 2 is the influx of calcium. So that's where the calcium enter into the cell to activate the muscle contraction. Then the phase 3 is the efflux of potassium, which is the repolarization step. And the phase 4 is the restoration of the resting membrane potential. So it goes to the complete resting membrane potential. Okay. So these are all the 5 different steps. So this kind of action potential develops in the working cardiac myocytes and not in SA nodal cells or not in AV nodal cells. Okay. If you look at the SA nodal cells, which is a natural pacemaker cells, and these cells has a different type of action potential. So here, the action potential is entirely different from the working cardiac myocytes, and this is mainly because of the composition of the cells. So if you look at the pacemaker cells, which are located in the SA nodal cells, they contain mostly glycogen rather than the contractile apparatus. Okay. And these cells also express different type of potassium and sodium channels, and therefore these cells has no true resting membrane potential. Unlike the working cardiac myocytes, for example, if you take a working cardiac myocyte, it undergoes a depolarization followed by a repolarization, and then finally it goes to a resting membrane potential. But in case of the SA nodal cells, there is no true resting membrane potential because of different, activation of different set of the ion channels. So let's see what activates the membrane depolarization and repolarization in SA nodal cells. So in the case of the pacemaker cells or the SA nodal cells, the invert calcium current or T-type calcium current is the one which depolarizes these cells. Unlike the sodium channels in a working myocytes, here the T-type calcium channel will depolarize the resting membrane potential. And then followed by the activation of the rectifier potassium current, which will repolarize the cells. And before it goes to the resting membrane potential, and then there will be another current called funny current, which is an invert sodium current, which is specific for SA nodal cells, which will destabilizes the resting membrane potential. So it undergoes the depolarization immediately. So why it is important? So because of this, quick activation, you will see a continuous stimuli of the heart rate. So for example, this entire system works, 60 to 70, cycle per minute. So which is corresponding to, 60 to 70 heart rate. And each channel, the T-type calcium channel and the potassium channels and the funny current, they are all activated by catecholamines and acetylcholine. So which represents the increased heart rate during exercise or during stress conditions, you will have an increased heart rate. Or during resting conditions, you will see a decreased heart rate. These are all maintained by these ions, which are all responding to the beta-adrenergic receptor activation. So we'll see that how these the T-type calcium channels, funny currents are activated during catecholamine stimulation and acetylcholine stimulation in one of the slides. Okay. So now we know, like, how the cardiac myocytes are stimulated, the action potential develops. And then, as I said, the calcium is the major, ion which stimulates the muscle contraction. So in a resting condition, the calcium channels are not activated. However, once the action potential develops, the calcium channel get activated, and which triggers the muscle contraction. So the next few slides, we are going to see how the calcium stimulates the muscle contraction. Okay, so before we go into the contraction relaxation cycle, I just wanted to introduce two terms. The first one is diastole. So diastole refers to the ventricular relaxation. Okay, wherever you see diastole or diastolic contraction, that indicates the ventricular relaxation. And then the systole is the ventricular contraction, systolic phase or systole. So that refers to the contraction of the muscle. So both the diastole and systole depends on calcium concentration inside the cell. So the calcium concentration within the cytoplasm, modulates, whether the cell has to go through the contractile phase or the systolic phase or the diastolic phase. So the next few slides, we're going to see how the calcium is important and how it is modulating the diastole and systole. So if you look at this cartoon, it shows like all the major calcium channels in the plasma membrane as well as in the sarcoplasmic reticulum. So as I said in the beginning, the plasma membrane plays an important role because the plasma membrane also forms a structure called T-tubules. So T-tubule is nothing but invagination of the plasma membrane, which goes deep inside the cell. And the T-tubule express large number of the L-type calcium channel, which is also referred here as DHPR. DHPR is dihydropyridine receptor, which is another name for the L-type calcium channel. And then the other calcium regulatory proteins are plasma membrane calcium ATPase and sodium calcium exchanger. And these two also plays an important role in maintaining the intracellular calcium. And most of the calcium inside the cells is stored in the lumen of the sarcoplasmic reticulum. So the sarcoplasmic reticulum is a membranous structure. Again, it is a modified form of endoplasmic reticulum, and it has a different type of proteins compared to the endoplasmic reticulum. And the major proteins are the ryanodine receptor, which is the calcium release channel, so which releases the calcium from the lumen of the SR to the cytoplasm. And then the SR calcium ATPase, which is referred here as a SERCA2A, which is a calcium pump which sequesters the calcium into the lumen of the SR. So this is a calcium release channel, and the SERCA is a calcium uptake channel. So these two plays an important role in maintaining the cytoplasmic calcium as well as the sarcoplasmic reticulum calcium concentration. Okay. And if you look at the small box here, it shows the calcium concentration in different conditions. So, okay, if you look at the sarcoplasmic reticulum, which has almost like 500 micromolar calcium. So it has a large amount of calcium stored in the lumen of the SR or the sarcoplasmic reticulum, which is around 500 micromoles. And then the outside the cell, which is the extracellular matrix, which has 2 to 3 millimolar calcium. It's a normal calcium concentration. And then if you look at the systolic calcium, which refers to the calcium levels in the cytoplasm during cardiac contraction, which is 1 micromole. Whereas in the diastolic calcium concentration is 0.1 micromole. So what it suggests is you need high concentration, large amount of calcium in the cytoplasm for its contraction, whereas the calcium needs to be removed from the cytoplasm to initiate the muscle relaxation. So the calcium needs to be cycled during contraction and relaxation in a proper way. And if there is a defect in calcium cycling, that will end up in calcium, the contractile defect and cardiomyopathy. So during contraction, so the cytosol should contain 1 micromole, and this calcium comes from the sarcoplasmic reticulum. And then during muscle relaxation, which is a diastolic phase, this calcium will be removed from the cytoplasm, and it will be restored in the endoplasmic reticulum or the sarcoplasmic reticulum, and some will be removed out of the cell. We're going to see what proteins are involved in this process. So, I told already like what is EC coupling or excitation contraction coupling. So once the action potential develops, and then the calcium channel get activated, and this calcium channel is a L-type calcium channel, which is also shown as DHPR in the previous slide. Okay, so the DHPR or the L-type calcium channel is the one which get activated when the membrane is depolarized. Once the action potential develops, the L-type calcium channel is, get activated, and it brings us a small amount of calcium from the extracellular matrix to the cytoplasm. Okay, so most of these L-type calcium channels are located in the T tubules, which is which is the invagination of plasma membrane or the sarcolemma. And once a small amount of calcium enters into the cytoplasm, this calcium will trigger the calcium, which is stored in the sarcoplasmic reticulum by stimulating the ryanodine receptor. So the ryanodine receptor is the calcium release channel. And once the small amount of calcium comes from the L-type calcium channel, which will trigger the ryanodine receptor, so then the calcium, comes from the sarcoplasmic reticulum to the cytoplasm. Then that calcium will binds to the contractile protein, which is troponin C, and stimulates the actomyosin interaction. And once the muscle contraction started, then the calcium will be taken back into the sarcoplasmic reticulum by SR calcium ATPase or SERCA pump, and then the muscle will start to relax. So this is the cycle, the calcium cycle associated with muscle contraction. So this process is also called calcium-induced calcium release, because you need a small amount of calcium to stimulate the calcium release from the sarcoplasmic reticulum. Therefore, it is called calcium-induced calcium release. And as I said, the different levels of calcium is important for contraction and relaxation. So the calcium-induced calcium release is mainly controlled by the L-type calcium channel and ryanodine receptor. And these two are called as receptors because the L-type calcium channel binds to dihydropyridine. Therefore, it is called as dihydropyridine receptor. And the ryanodine receptor is the one which binds the ryanodine. That's for, it has no clinical relevance, but since these compounds binds to these receptors, or to these channels, it's called as receptors. Okay, so the calcium-induced calcium release is mainly controlled by the ryanodine receptor and L-type calcium channel. So why it is important? So if you look at the previous image, this is the L-type calcium channel, which is located in the T tubules, and this is the ryanodine receptor, which is located in the sarcoplasmic reticulum. So they located in a very close vicinity. So once the L-type calcium channel is activated, it will stimulate the ryanodine receptor simultaneously to release the calcium, so the muscle contraction will start. So the L-type, so the T tubule, therefore, has a very important function in calcium-induced calcium release because it is involved in excitation contraction coupling because the T tubule increases the surface of the plasma membrane. So unlike any other cell types, you can see a huge amount of plasma membrane in cardiac myocytes, mostly in the form of T tubules, which goes deep inside the muscle cell. And this increase in the surface increases the density of L-type calcium channel, and therefore it speeds up the excitation contraction coupling. And it is also a receptor coupling. Wherever L-type calcium channel is there, you will see the ryanodine receptor in a close proximity. So these two receptors, they kind of couple together functionally and speed up the calcium entry. And then the third is SR coupling. So the T tubule will kind of synchronize the calcium release from the sarcoplasmic reticulum by stimulating the renin receptor. Because of these, three important functions, the T tubules play a major role in calcium-induced calcium release. So during muscle contraction, the calcium enters into the cell, or the calcium influx, by two different mechanisms. One is through the L-type calcium channel, and then the second is through the renin receptor. And these two channels are, as I said before, are called receptors because they bind to specific drugs. So, these two channels are involved in muscle contraction. And what is the mechanism behind the muscle relaxation? So the calcium removal is the major mechanism by which the muscle relaxation occurs. And the calcium is removed from the cytoplasm by four major proteins. The first one is sarcoplasmic reticulum calcium ATPase. I mean, this cartoon basically shows how the calcium is cycled inside the cell during muscle contraction and relaxation. So once the action potential develops, the calcium enters and it stimulates the calcium release through the renin receptor. And then this calcium goes and binds to the troponins T and then stimulate muscle contraction. Then the calcium is taken back into the sarcoplasmic reticulum. So it removes the calcium, and some other calcium will be taken up by mitochondria, and some calcium will be removed by sodium calcium exchanger and plasma membrane calcium ATPase. So by this mechanism, the calcium is cycled within the cell and also outside the cell. So the major mechanism by which the calcium is removed is by sarcoplasmic reticulum calcium ATPase, or SERCA pump, and this pump restores the calcium into the SR, and this mechanism represents almost like 90% of the calcium reuptake process. And again, it is working against the gradient because the lumen of the SR itself contains like 500 micromolar calcium. So the pump has to work against the gradient. Therefore, it requires energy, and the energy is provided in the form of ATP. The pump itself is an ATPase, so that's why it is called a sarcoendoplasmic reticulum calcium ATPase. So by using the ATP as energy source, it restores the calcium into the lumen of the SR. And the activity of this pump is regulated by a protein called phospholamban. The phospholamban is an inhibitor of SERCA pump, and however, the phospholamban is phosphorylated by pKa or CAM kinase, and then the phospholamban will relieve its inhibitory effect, so the pump will be more active. So this is one of the major mechanisms by which the contraction is increased during catecholamine stimulation, particularly when you are doing exercise or during stress conditions. Even in heart failure, you will see the increased levels of catecholamine, and the catecholamine will activate kinases, particularly pKa and CAM kinase, and this is one of the mechanisms by which it will speed up the muscle relaxation. And the second mechanism is through the sarcolemmal sodium calcium exchanger. Here, the calcium will be exchanged for sodium ions. So it will exchange one calcium for 3 sodium. And this represents 5% of the calcium removal. So 90% of the calcium is removed by sarcoplasmic reticulum, which is restoring the calcium back into the SR for the next contraction cycle. But 5% of the calcium will be pumped out, and thereby it will bring some sodium ions. And as I said before, sodium is important for muscle repolarization. So, sodium calcium exchanger also plays an important role in muscle repolarization. And 2% of the calcium is taken by the mitochondria using a mitochondrial calcium inhibitor, and the calcium will speed up the ATP synthesis. So, the calcium will activate several enzymes involved in oxidative phosphorylation, and so thereby it will increases the ATP production. And 1% of the calcium is removed by the plasma membrane calcium ATPase, also called as sarcolemmal calcium ATPase. That's how, like, it removes almost all the calcium from the cytoplasm during muscle relaxation. Okay. So this we have already seen how the calcium, now we're going to see how the calcium is stimulating the actomyosin interaction. So we have already seen this, like the thick filament and thin filament. So thick filament contains myosin. So the myosin contains myosin heavy chains and light chains. And thin filament contains actin, tropomyosin, and troponin complex. And the other proteins are all involved in regulatory role, whereas the thick myosin and actin are involved in muscle, the contraction and relaxation. So the next few slides, we're going to see how this actomyosin interaction works. So the first important molecule is the myosin, which is a molecular motor. It's a dimeric molecule containing a filamentous tail and a globular head. And this head contains actin binding site. Also, it has the ATPase activity. So this is where the ATP binds and hydrolyzed. And also, this is the region where it interacts with the actin molecule. So the myosin contains two myosin heavy chains and four light chains. And four light chains are classified into two types. The myosin light chain 1. So it has a pair of myosin light chain 1 and a pair of myosin light chain 2. So the myosin light chain 1 is called as the essential light chain, and light chain 2 is called as a regulatory light chain. So it can get phosphorylated. The phosphorylation will enhances the kinetics of actomyosin interaction. The second important protein is actin, which is a small globular protein where it forms a filamentous actin, and it interacts with myosin, and thereby, it slides over the myosin head and resulting in muscle contraction. See, the other thin filament proteins like tropomyosin and troponin are mostly involved in regulatory mechanisms. For example, the tropomyosin usually prevents the actomyosin interaction in a relaxation state. When a muscle is in a relaxation state, the tropomyosin will binds to the myosin binding site in the actin filament, and thereby it prevents the myosin interaction. The troponin complex, it has three different proteins: troponin C, T, and I. The C stands for calcium. So this protein binds to calcium and activate the contractions. So whenever the calcium-induced calcium release mechanism occurs, the calcium will bind to troponin C, and thereby it will activate the actomyosin interaction. So the binding of calcium to troponin C is therefore involved in muscle contraction. The troponin T is involved in moving the tropomyosin, which prevents the actomyosin interaction, and thereby it releases its inhibitory effect. Once the calcium binds to troponin C, the entire complex, the troponin complex, will be rearranged, and thereby the troponin T will kind of move the tropomyosin away from the actin, and thereby it exposes the myosin binding site to actin molecule. And troponin I regulates the calcium release, and thereby it initiates the muscle relaxation. And among these three, the troponin I is a phosphorylatable protein. So if it is phosphorylated by pKa, and it can increases the muscle relaxation. So, the kinases will phosphorylate, particularly the pKa will phosphorylate several proteins, and troponin I is one of them, which is important for the catecholamine-mediated increased muscle contraction and relaxation. Okay, so probably you might have studied this also, the actomyosin interaction, which is explained by cross-bridge cycling. Cross-bridge represents the attachment of myosin head to the binding site on the actin filament, and the repetitive attachment and detachment of myosin head to the actin, filamentous actin, is called as cross-bridge cycle. So initially this was proposed as a theory, but then later on it has been proved. there are a lot of multiple small steps in the actomycin interaction. We're going to see the major steps involved in the actomycin interaction in a cross-bridge cycle. So in the presence of calcium, once the calcium is released from the sarcoplasmic reticulum by action potential development, the calcium will bind to troponin C and followed by the actomycin interaction. So in this case, it is a generalized step. So once the calcium is there, the myosin head will bind to the actin. So as I said before, the myosin itself is an ATP, so it hydrolyzes the ATP to ADP. And so you will see ADP and phosphate molecule in the pocket of the myosin ATPase, which will not be released until the power stroke develops. So once the power stroke comes in, then the myosin head will bend back, and thereby it will generate a force, and it kind of slides the actin molecule over the head. And during that step, the phosphate and ADP molecule will be sequentially released. So first the phosphate molecule will be released, followed by the ADP molecule. So once these molecules are released, then the head will undergo a low energy configuration, and the de-energized head then binds the next ATP molecule. Then it is ready for the next contractile cycle, the actomycin interaction cycle. So this is how the cross-bridge cycle works in a in a simple way, but in a detailed molecular aspects. So during diastole, during muscle relaxation, the ATP binds to myosin. However, it cannot interact with actin molecules because the tropomyosin, prevents the actomycin interaction. Since the myosin is ATPase, it immediately hydrolyzes the ATP, but the ADP and phosphate, they remain in the nucleotide pocket until the calcium stimulation occurs. Once the calcium stimulation occurs, the troponin complex, the troponin T, will move the tropomyosin, and followed by the phosphate release from the pocket, and thereby the actomycin interaction occurs. So which is actually shown here. You can see this is the actin, this is the myosin binding site, and this is the myosin head. So in a normal conditions, the tropomyosin will kind of prevent the myosin head to interact with the myosin binding site. But once the calcium comes in, the entire complex will kind of change its conformation and exposes the myosin binding site of the actin to the myosin head. So the myosin head will interact with actin molecules. So once the phosphate is released, then the myosin head will de-energized and bends its head, and thereby it creates a power stroke, and that slides the actin molecule. This change in conformation will release the ADP molecule. Then the next ATP binds and the myosin head, and then the relaxation starts. So if the calcium is present, then it will undergo the next contraction cycle. If there is no calcium, then it will stay in the relaxation stage. So this is how the cross-bridge cycle works. Okay, so the next important topic is metabolism. So why cardiac metabolism is important? Because the cardiac muscle requires large amount of energy for its repetitive contraction and relaxation. And this is also evident from the dense vascularization and abundance of mitochondria, as I said before. So if you measure the individual organelles in a cardiac myocyte, 30% of the cell volume is occupied by mitochondria, suggesting that you need large amount of ATP molecule, which will be provided by the mitochondria. And also you will see a large vascularization. If you take a section of the heart, stain it for the the blood vessels, you will see a dense microvessels, suggesting that you need oxygen also for the ATP generation. So most of the energy or the ATP molecule comes by oxidative phosphorylation. And the heart consumes a vast quantity of energy. It cycles almost like 6 kilogram of ATP per day, because that much amount of ATP is synthesized and used. If you measure the ATP levels, you may not see any ATP at all, because that will be consumed very quickly by the cardiac muscle. So the most of the energy is come from the oxidation of fat and carbohydrates, and also some extent from other sources like proteins and amino acids. And the adult heart prefers fatty acid. Okay. And the neonatal heart and embryonic heart, they uses carbohydrates as a primary energy source. And the energy made from a given amount of oxygen is similar for all the substrate, because fatty acid, although it gives more calories, but it requires more oxygen for its oxidation compared to the carbohydrates. So therefore, the cardiac efficiency is usually calculated based off energy equivalent of oxygen consumed. So why this is important? In many of the heart failure, you will see a decrease in cardiac efficiency, because the most of the energy will be used by the failing myocardium for its function. However, the energy synthesis will be less, so there will be an imbalance between energy production and usage that will reduce the cardiac efficiency. So what determines the cardiac metabolism? There are three main factors which determines the cardiac metabolism. The first one is workload, and the second is substrate, and third is oxygen. So cardiac myocyte has a very high metabolic rate. As I said before, it consumes large amount of ATP molecule, and also it's synthesized quickly and used it because it contracts repetitively. So the cardiac muscle contracts 1 to 3 times per second, depending on the conditions, from resting condition to the exercise. And most of this energy, which is synthesized in the form of ATP, is used by three important molecules, which we have already seen. The first one is myosin. So the myosin uses 50% of the ATP molecule synthesized by the cardiac muscle, myocyte. And then the SR calcium ATPase uses 25% of the ATP molecule synthesized by the cardiac myocytes. And sodium potassium pump, or sodium potassium ATPase, which is involved in action potential, uses 15% of the ATP. So if you compare, all three, most of the energy which is produced by the cardiac myocytes are used by myosin, SERCA, and sodium potassium ATPase. Only the remaining ATPase is used for other cellular functions. So therefore, if you increases the workload, the myosin activity will go up, and SERCA pump activity will go up, and your action potential development will also go up. So meaning it's going to use more and more energy for all these functions. So depending on the workload, the energy use will therefore go up. So from resting conditions to the exercise or during pathological conditions. And then the second one is substrate. So depending on the substrate availability, the energy production will be different. For example, if you have fatty acid, you're going to produce more ATP molecules compared to carbohydrates. However, the cardiac muscle is metabolically omnivore, meaning that it can utilize any type of energy which is available in the system. Even if the fatty acids and carbohydrates are not available, it'll use as like, the amino acids and other molecules to produce ATP molecule. So the two main substrates are fatty acids and carbohydrates or glucose. Fatty acids are mostly used during the fasting conditions and glucose in fat conditions. And the heart also uses lactate during exercise. If these two levels goes down, it will take the lactate from the blood, and then it will convert to pyruvate and use it. So the substrate is therefore another important determinant of cardiac metabolism. And then the third one is oxygen. So the sufficient supply of ATP can be achieved only through the oxidative phosphorylation, which requires aerobic respiration. However, in some conditions, for example, oxygen deprivation conditions like ischemia, and the heart will be kind of converted to anaerobic metabolism, although it provides less amount of ATP molecules. So whether the ATP is, the oxygen is available or not, the system will kind of switch over to produce ATP molecule. Even in some conditions, it will break the ATP molecules and produce like ATP and AMP molecules to supply the myosin circuit and sodium potassium ATPase. So these three factors are therefore the important determinants of cardiac metabolism: workload, substrate, and oxygen. So depending on the changes, the metabolic system will be altered. So the first one is glucose metabolism. We'll quickly go over how the glucose is metabolized in the cardiac muscle. Like any other cell types, the glucose will, concentrate. If the glucose concentration is high in the circulating blood, the glucose utilization will be stimulated by insulin, and the glucose will enter into the cell through the glucose transporters. The cardiac muscle expresses, GLUT1 and GLUT4. GLUT1 is a ubiquitous form. It expressed in all cell types. And GLUT4 is called as insulin dependent. And usually GLUT4 is stored in the endosomes. And once the glucose level goes up, the insulin will stimulate the GLUT4 to recruit it to the plasma membrane, and thereby it will enhances the glucose utilization. So both GLUT1 and GLUT4 will be recruited more to the plasma membrane when the glucose level goes up, and then thereby it will bring the glucose inside. Once the glucose enters inside, it will be phosphorylated to glucose 6-phosphate. Thereby it will prevent the glucose to exit the cell, to prevent the glucose to go out of the cell. It will be phosphorylated. Once it is phosphorylated, it cannot penetrate the plasma membrane. So then the glucose 6-phosphate will undergo a series of reactions to produce pyruvate molecule, and then the pyruvate will enter into converted to acetyl-CoA and enter into the mitochondria, where it will undergo Krebs cycle and electron transport and produce ATP molecule. And some of the glucose will be stored in the form of glycogen, which will be used by the cardiac muscle during exercise. The glycogen will be mobilized mostly depending on the substrate availability. If you have more fatty acid, like the glucose will be converted to glycogen and stored. And then once the fatty acid level goes down, then like the glycogen will be broken into glucose 1-phosphate and then to glucose 6-phosphate and enter into the glycolytic pathways. Some of the glucose will be, undergo the pentose phosphate pathway. It will produce the 5-carbon sugar, which is important for nucleic acids and some of the signaling system also. So what is important here is the regulatory steps. So this is the entire glycolytic pathway. So glucose 6-phosphate is converted to fructose 6-phosphate and then to fructose 1,6-bisphosphate by phosphofructokinase. Then the 6-carbon sugar is broken down into 3-carbon dihydroxidane and glyceraldehyde 3-phosphate. Then the glyceraldehyde 3-phosphate will be converted to pyruvate through a few steps. And some of the steps are important. For example, the glucose transport. So the glucose transporters are regulated depending on the availability of the glucose in the blood. And the glucose phosphorylation through hexokinase, which is also regulatable. So depending on the workload, these kinases will be phosphorylated, and these kinases will be activated by the substrates. Most of these enzymes are allostatic enzymes. So depending on the substrate levels, these enzymes will get activated, and thereby it will enhances the activity of this entire glycolytic pathway. So then hexose phosphate phosphorylation through phosphofructokinase, which converts the fructose 6-phosphate to fructose 1,6-phosphate. This is also one of the regulatory steps in cardiac muscle. And then the triose phosphate oxidation through glyceraldehyde 3-phosphate dehydrogenase, pyruvate oxidation through pyruvate dehydrogenase. All these are all important regulatory steps which would get activated during exercise or catecholamine stimulation, as well as these steps are also modified during pathological conditions. The second important substrate is fatty acid. The fatty acid concentration is very high in the circulatory system. The fatty acid will bind to the proteins and enter into the cell through the fatty acid transport protein, also called as CD36. Then inside, the fatty acid will be converted to fatty acyl-CoA. Then it will enter into the mitochondria through carnitine palmitoyl transferase, or CPT. Inside the fatty acid CoA undergo fatty acid oxidation. Then it converted to acetyl CoA, then enters the TCA cycle, and then electron transport chain to produce ATP molecule. So the most important regulatory step is the carnitine palmitoyl transferase, which transfers the fatty acid from cytoplasm to the mitochondria. And this step is also accelerated during catecholamine stimulation to increases the energy production. Okay, so the most important one is a substrate interaction. So as I said, the cardiac muscle is metabolically omnivore, so it can utilize any type of substrate available. So this cartoon illustrates how these cardiac muscle behaves depending on the substrate availability. So for example, in a fasting stage, you're going to have a very low amount of carbohydrates, and during that step, the fasting will stimulate the free fatty acid production through the lipolysis of the adipose tissues, and then the fatty acid will be utilized by the cardiac myocytes. So during fasting state, fatty acid is the major energy source for the heart. And then whatever the glucose comes in at that time will be minimally utilized, or it will be stored in the form of glycogen. So whereas in a fed state, when the glucose level or the carbohydrate level goes up, then the insulin level goes up, and then the insulin will stimulate the glucose utilization. At the same time, the insulin will also shut down the fatty acid production. So it will kind of reduces the lipolysis from the adipose tissues. So the free fatty acid level goes down, and then there won't be any fatty acid uptake. So it is not 100%. So, there is always a substrate interaction. So this is not like 40-60 or 100-0%. If you have 50% of carbohydrate, 50% will be from the, fatty acid. Or if you have 60%, 30% lipid, and then 60% will be carbohydrate. So there will be always a, substrate interaction, and it is not an all or nothing phenomena. So depending on the availability of the substrate, the heart will utilize this to produce ATP molecule, because the heart needs continuously ATP supply for its function. So if there is any kind of disruption in the metabolic activity, that will contribute to cardiac pathology. Also, people have shown that during cardiac pathology, there is a defect in metabolic pathways. So for example, as I said before, the normal heart, it will utilizes fatty acid or carbohydrates depending on the substrate availability. However, if the heart undergoes a mechanical stress, mechanical overload, you will see a decrease in fatty acid oxidation. However, the glycolysis will go up. So during this conditions, during mechanical overload, you will see increased glycolytic pathways. So, but however, the glycolytic pathways will produce only less amount of ATP molecule compared to fatty acid. So at the end, you will see a decrease in energetics. And if it is not treated properly, then you're going to see defect in oxidative metabolism, increased oxidative stress, and then insulin resistance, lipid accumulation, energy deprivation that results in heart failure. So And this has been proved in animal models and human. In heart failure conditions, you will see a decrease in oxidative metabolism, increased oxidative stress, lipid accumulation, insulin resistance, all those things. And now, like, even targeting the metabolic pathways is one of the, method for treating the heart failure patients. So this entire process is called as metabolic remodeling. And this kind of remodeling can contribute to the heart failure, or it could be like, part of the heart failure process. In the case of obesity and diabetes, it is exactly opposite, like the fatty acid oxidation goes up and the glycolytic oxidation, glucose oxidation goes down. And if it is not treated properly, you will see a decrease in the efficiency. And again, it undergoes, oxidative metabolism and oxidative stress and insulin resistance, all those things. So, in general, in pathological conditions called as pathological hypertrophy, in response to mechanic overload like hypertension, valve disease, myocardial infarction, you will see decrease in fatty acid oxidation and increases in glycolysis. And this is also called as fetal-like metabolic profile, because as I said before, during development as well as in neonatal conditions, the glucose is the preferred substrate for the cardiac muscle. So during stress conditions or during pathology, you will see a reversal of the same conditions. The heart will start to use glucose molecules than the fatty acid. So that's where it's called as fetal-like metabolic profile. So this will decrease as the ATP synthesis, and then finally you will end up with energy deprivation and starvation model. Okay. So now we are moving on to the signal transduction. We're going to focus on a specific signaling molecule, which is a catecholamine-mediated signaling. So in cardiac muscle, you can see all type of signaling mechanisms. So whatever you have studied in other cell types, you can see the same mechanism in cardiac muscle. But however, the one which is specific for cardiac muscle is a catecholamine-mediated signaling. So the catecholamines are the major mediators of the cardiac response to increased workload. Like when you do exercise, and also in heart failure conditions, you will see increased levels of catecholamines, and these catecholamines will stimulate a specific receptor called beta adrenergic receptor. So what are catecholamines? So catecholamines are synthesized from the amino acid tyrosine. So there are two types, epinephrine, which is also called as adrenaline. It is released by the adrenal medulla. So it has a systemic effect, and it binds to beta adrenergic receptors and then stimulates the heart. And it has a global cardiovascular effect. And then the second one is norepinephrine. It's also called as noradrenaline. It's a paracrine factor. It is released by stellate ganglion, which is located at the level of seventh cervical vertebrae. And it also binds to beta receptors. I mean, the epinephrine and norepinephrine mostly binds to beta receptors, but also binds to alpha receptors with a different affinity. But for current purpose, like, we're going to focus only on the beta adrenergic receptors, how the epinephrine and norepinephrine will stimulate the beta receptors and activate the signaling molecules. So the catecholamine activates a G protein coupled receptors. So the beta receptors are G protein coupled receptors. And once the catecholamine level goes up due to exercise or during heart failure conditions, you will see increased levels of catecholamines. And the catecholamine will bind to beta receptors, and the beta receptors will activate a specific set of G proteins. And this G protein is a GTPase, meaning so it spontaneously hydrolyzes the GTP molecules. So this G protein has three subunits, alpha subunit, gamma and beta subunit. The one which is associated with the catecholamine is called as alpha stimulatory subunit or alpha S. In other words, you can also see stimulatory alpha subunit. So you will see a stimulatory alpha subunit and a gamma subunit and beta subunit. And in a normal conditions, the alpha subunit hydrolyzes the GTP molecule, and keeps the GDP. And this GDP complex is an inactive complex. Okay. So once the beta receptor is stimulated, and the GDP will be replaced by GTP molecule. So once the GTP is bound to the alpha subunit, the alpha subunit will separate from the beta gamma complex. And then the alpha subunit will bind to another enzyme called adenylyl cyclase. So the adenylyl cyclase or adenylate cyclase will stimulate the cyclic AMP production. Cyclic AMP is one of the second messenger, and then the cyclic AMP will activate the protein kinases for its function. So once the activation occurs, then the GTP will be hydrolyzed and it becomes a GDP. Then it will relocate to the alpha beta complex and becomes inactive. So this is how the entire catecholamine system works. So once the beta receptor activated, then it activates the adenylyl cyclase. The adenylyl cyclase converts the ATP to cyclic AMP. Cyclic AMP is the second messenger. The cyclic AMP, one of the the cyclic AMP activates like several things, like including kinases and transcription factors. One of them is PKA. PKA has a major role in cardiac contraction during catecholamine stimulation. So the cyclic AMP binds to a protein kinase A, which has two regulatory subunits and two catalytic subunits. So in the absence of cyclic AMP, the regulatory subunit binds to the catalytic subunits and becomes inactive. Once the cyclic AMP binds to the regulatory subunit will detach from the catalytic subunit, and then the catalytic subunit is the active protein kinase A. So protein kinase A phosphorylates several proteins, and I'm going to show you some of the important proteins which are involved in muscle contraction. Okay, so the first one is it's going to phosphorylate the funny current, which is involved in the action potential in SA nodal cells. So this one will increases the heart rate. So the heart rate will be faster. If you run or if you do exercise, your heart rate will increase. That's mainly because of the activation of the funny current, and this is called as positive chronotrophy. So positive chronotrophy means enhanced heart rate. So if you see negative chronotrophy, that means reduced heart rate. So PK, one of the target is the funny current, which increases the heart rate. And then the second one is L-type calcium channel. So we have already seen L-type calcium channel is involved in calcium-induced calcium release during action potential development and muscle contraction. So the phosphorylation of L-type calcium channel will increases the opening probability of L-type calcium channel. Therefore, the contraction will be speeded up. So you will see an increased contraction, which refers to ionotrophy. So increased ionotrophy means increased muscle contraction. In addition to L-type calcium channel, the PKA can also phosphorylate one of the light chains, which is a myosin light chain 2 in the myosin molecule, which will enhances the kinetic of muscle contraction. That will also contributes to increased ionotrophy. The PKA at the same time can also phosphorylate phospholamban, which is an inhibitor of SR calcium ATPase. So once the phospholamban is phosphorylated, the SERCA activity will go up. So it will quickly restore the calcium. So the muscle relaxation will be faster, which refers to increased lucidrophy. And troponin I said before, like the troponin I is an inhibitor of, the it actually activates the tropomyosin. And once it is phosphorylated, it's going to increases the the lucidrophy or the relaxation. So both phospholamine and troponin high phosphorylation will increases the lucidrophy. So by PKA activation, you will see an increased heart rate and you will see an increased rate of contraction and increased rate of relaxation. So your heart will, function much faster and the pumping will be much faster too. So in addition to that, the PKA can also improve the glucose transport, and it also enhances the production of fructose 2,6 bisphosphate. It is one of the activator of glycolytic pathway, and it also enhances the glycogen synthase as well as glycogen phosphorylase. So the entire glycolytic pathway will get activated to produce more ATP molecule. The long-term activation of PKA can also activate the transcription factors. For example, it can activate cyclic AMP responsive element binding protein CREB, which is a transcription factor which can activate several genes involved in muscle contraction and enhances the muscle mass, which is referred to as hypertrophy. So the catecholamine stimulation, therefore, not only increases the rate of contraction and relaxation, but also the ATP synthesis and also the protein synthesis in the cardiac muscle. Okay, so we have seen so far the catecholamine stimulation, but what happens in a resting condition? So what controls the resting heart activity? So which is mainly by acetylcholine. So the acetylcholine is an antagonist of catecholamine. So it will do exactly the opposite of whatever the catecholamine does. So it will reduces the cyclic AMP synthesis, it will reduces the PKA activity, so everything will be reversed. So it blocks the effect of catecholamines on cardiac contraction. So therefore, you will see a negative inotrophy. And then it will also reduces the heart rate. And so you will see a negative chronotrophy. So it will exactly the opposite of catecholamine does. So the mechanism is also somewhat similar. So the acetylcholine will be released by vagus nerve, and it binds to a specific receptor called M2 receptor in cardiac myocytes. So acetylcholine binds to different tissues in different receptors, but in cardiac muscle, it binds to specifically M2 receptor, and the M2 receptor is coupled to another set of G protein. Here, the G protein complex is called as inhibitory G protein. So in the case of catecholamine, it's called stimulatory catecholamine G protein, and here it is an inhibitory complex. So again, it has three different complexes, the alpha subunit, beta, and gamma subunits, and the alpha subunit is an inhibitory subunit. But the beta and gamma subunits are similar to the one which is shown in catecholamines. So once the M2 receptor is activated, the GI will get activated, and then the GI will stimulate or it will go and binds to the adenylyl cyclase and prevents the cyclic AMP production. So at the same time, the beta gamma complex can also open the potassium channel in the SA node. So that will inhibits the spontaneous depolarization, and thereby it reduces the heart rate. So it reduces the PKA activity, also the heart rate. So your entire heart rate and cardiac contraction will go down. So this is in resting conditions. Okay. So the next two parts, we're going to see the basic aspects of heart diseases, what causes the heart failure or heart diseases, and then we're going to see one example, which is myocardial ischemia. Okay, so the cardiac muscle or cardiac myocytes responds to stress in a different way compared to other organelles. This is mainly because the cardiac myocytes are terminally differentiated and the myocyte cannot further undergo cell division. So, if you, for example, if you take liver, if you chop off some liver, the liver will grow back because of the because of the hyperplasia or cell division. Whereas in cardiac myocytes, the myocytes are terminally differentiated. so they cannot further divide. So if you look at the cardiac development, the cardiac myocytes proliferate during embryonic conditions and maybe one day after birth. Then after that, the myocyte growth or the myocyte will grow only by enlarging its size, not by cell division, by but by increase in its size. That's called as hypertrophy. So hypertrophy is therefore refers to a mechanism by which the cell increases in size, which is different from hyperplasia, where the cell divides. So in the adult heart, the cells are terminally differentiated. So if you stress the heart, and then the heart has to work more. So instead of dividing the cells because it's terminally differentiated, these myocytes undergo hypertrophy and they become enlarged in its size. So this, hypertrophy is mainly to increases the contractile capacity of the myocytes. So if you look at the hypertrophied myocytes, you will see more sarcomeres, and also the sarcomeric length will go up. I showed you before the sarcomeric length will be like somewhere few microns, and that will grow up because of the addition of sarcomeres, and also the myocyte width and length will increase from more than 100 microns. So this hypertrophic process, is mainly to improve the contractile capacity of the heart, but at the same time, since you are adding more sarcomeres, it requires more energy, more ATP molecules, because the sarcomeres composed of myosin ATP, which requires almost like 50% of the ATP synthesized by the cardiac myocyte. So if you add more sarcomeres, it requires more energy. So in a cardiac system, like if the muscle undergoes a hypertrophic conditions, it not only like synthesize more protein, but also it is like trying to produce more ATP molecules. So this hypertrophic process can occur in different conditions. So depending on that, if it is a pathological condition, it's called as pathological hypertrophy. And if it is a physiological modification, then it is called physiological hypertrophy. The best example for physiological hypertrophy are chronic exercise and also pregnancy. During pregnancy, the heart will enlarge, and that's mainly by physiological hypertrophy. And the physiological hypertrophy, sometimes it's reversible, meaning that it can go back to normal size. But however, the pathological hypertrophy is not reversible, it is irreversible. So the pathological hypertrophy can be caused by pressure increase or volume overload. So, those pressure and volume both can, cause pathological hypertrophy. So the difference between physiological hypertrophy and pathological hypertrophy is physiological hypertrophy, you will see enlargement of the cardiac myocytes, but there won't be any fibrosis and there is no inflammation, and you will see a normal increase in protein synthesis, normal vascularization and everything. In a pathological hypertrophy, you will see an increased fibrosis, necrosis, and then the contractile proteins are also kind of shifted towards fetal conditions. So, that's entirely different. So we're going to focus mostly on pathological hypertrophy, not on physiological hypertrophy. So the pathological hypertrophy is adaptive mechanism. So when you stress the heart is trying to increase its contractile capacity by hypertrophic process. So initially, it is an adaptive mechanism. However, if this condition is not treated, then it will undergo a maladaptive condition and becomes heart failure. Okay, so many of the heart failure conditions are caused by hypertrophy, pathological hypertrophy. So this is the diagrammatic representation of a normal heart and the hypertrophied heart. You can see a thick muscle, and the cavity will be very small, and you will see enlarged the myocytes at a cellular level. This is a human heart with a concentric left ventricular hypertrophy, and you can see the cavity is very, very small, the left ventricular cavity. Okay, so we're going to see first what causes hypertrophy. There are various stimuli can cause cardiac overload. We're going to see the major ones. The first one is hypertension. So you know what is hypertension is. So this is an increased stiffness in conducting vessels or in peripheral vasculature, or anywhere in the body, in a systemic circulation. If there is a resistance, and it's going to feedbackly affect the cardiac function. So that will result in enlargement of the heart. So you can see this is a normal human heart and this is a hypertensive heart. So the hypertensive heart is usually very, very big. So this type of hypertrophy is called as pressure overload induced hypertrophy. And then the second one is myocardial infarction. So myocardial infarction is mainly caused by the damage in the coronary artery circulation. So, for example, if there is an atherosclerosis in a coronary artery, and part of the heart will lose its oxygen supply, so those cells will start to die, and then that will be replaced by scar tissue. And to compensate the loss of a portion of the cardiac muscle, the remaining heart will become enlarged. So in case of myocardial infarction, the hypertrophy is mostly a volume overload hypertrophy, meaning like you'll see a large accumulation of blood inside the ventricle. And in hypertension, you will see the heart will develop a very high pressure to pump the blood. Okay, so valve dysfunctions. So we have seen this before, the aortic stenosis, aortic regurgitation. Those conditions can cause the cardiac overload. Usually the aortic valve stenosis causes a pressure overload hypertrophy, and aortic regurgitation causes a volume overloaded hypertrophy. And also the congenital diseases where you will see only like two leaflets instead of three in the aortic valve, something like this. So that can also causes the hypertrophy of the heart. The other congenital diseases are the abnormal communication between the chambers. So in the beginning, I said like there is no communication between the left and right part of the heart. This is prevented by the septum. So the atria, left and right atria are separated by atrial septum, and left and right ventricles are separated by interventricular septum. And if there is any kind of congenital malformation in these septum, that will causes the miscommunication between the left and right part of the heart that will resulted in hypertrophy. And in addition to that, the mutation in contractile proteins, for example, myosin binding protein C. So these two are the major contractile proteins involved in congenital diseases. The mutation in myosin or myosin binding protein C or troponin. So these protein changes can cause us hypertrophy, which is called hypertrophic cardiomyopathy. So this is also the common cardiac congenital heart diseases. Okay, so we'll see some of the examples of the congenital diseases. The first one is atrial septal defect. I mean, from the name itself, we know what it is. There is a defect in the atrial septum. So there is a miscommunication. I mean, there is a hole in the atrial septum. So the left and right atria, the blood will kind of send between left and right part of the atrium. So that causes a hypertrophy. And similarly, the ventricular septal defect, here there is a hole in the interventricular septum, so there is an abnormal opening between the left and right. So that results in a mixing of oxygenated and deoxygenated blood. So that can cause the hypertrophic conditions. And the third common one is tetralogy of Fallot. So here there are four defects in the heart. So one is the narrowed pulmonary artery, and then the abnormal position of the aorta. And since the pulmonary artery is narrow, it's going to cause enlarged right ventricle, the right ventricular hypertrophy, and also you will see a hole between lower heart chamber. The tetralogy of Fallot is one of the common birth defects, and it can be corrected surgically also. And the fourth one is patent ductus arterius. It's also called as PDA. This is an abnormal opening between aorta and pulmonary artery. So if you look at the anatomy, the aorta and pulmonary artery, they are all very close to each other. And during developmental defect, the aorta and pulmonary arteries, they fuse together. So there will be an opening. So that kind of causes the mixing of blood, and that results in hypertrophy. So what happens to the hypertrophic heart? So the hypertrophic heart, the main goal of the hypertrophy is to accumulate sarcomeric proteins, which is like contractile proteins, actin and myosin. So which is stimulated by several mechanisms. The first one is once the the extracellular stimuli is recognized by the cardiac muscle, if it is a pathological one, it's going to activate the sensors and receptors that will activate the signaling pathways. So one of the sensor is integrin that we have seen before, integrin, taline, and so the integrin signaling is one of the major pathological signal sensors which get activated during hypertrophic process. And then G protein coupled receptor activation, catecholamine stimulation, particularly in a heart failure conditions like, the catecholamine level goes up, it can also stimulate signaling pathways. So once this receptors and sensors are activated, it will activate the signaling pathways downstream, which includes like calcineurin pathway, which is a phosphatase, and also it activates the protein synthesis pathway like PI3 kinase pathway or MAP kinase pathways. And the activation of signaling pathways results in the activation of gene expression, and many of the genes will be, the contractile protein genes like myosin heavy chain, myosin light chain, actin proteins, and everything. And that will result in increased protein synthesis, so that will accumulate more sarcomeric protein. So this entire mechanism is adaptive during early stage of hypertrophic process. However, if it continues, you will see the activation of other mechanisms like fibrosis and necrosis. So the hypertrophy can also promote angiogenesis and fibrosis. So the first one is angiogenesis. So this is a normal myocardium, and you can see like normal myocardium is all connected with capillaries. And if there is a hypertrophy, the hypertrophied myocardium and the myocytes will kind of push the capillaries far away. So to compensate that, there you will see a new capillary formation, which is called as neocapillarization or angiogenesis, which is, to improve the oxygen supply to the hypertrophied myocardium. And this kind of neocapillarization is unique for pathological hypertrophy. In the case of physiological hypertrophy, for example, if you're doing a chronic exercise, you will see a lateralization. So the blood vessels will fuse together to improve the circulation. It's different from the pathological hypertrophy. And then the interstitium will be filled with collagen to produce interstitial fibrosis. So if you remember, I said, 70% of the cardiac muscle is occupied by non-cardiac muscle cells, which include fibroblast, endothelial cells and vascular tissues. So these fibroblasts, they will try to kind of convert to myofibroblasts to support the myocardium during pathological conditions. And the fibroblasts produce large amount of collagen. Collagen gives better tension to the myocardium. So it will enhances the muscle contraction, but the collagen cannot relax like a myocyte. So because of that, you will see a improved contraction during the initial phase of myocardial hypertrophy. But if it continues, you will see a improper relaxation causing a diastolic dysfunction. Okay. So this is one of the mechanism for the heart failure in pathological conditions. Okay, so if the hypertrophy is not treated properly, you'll undergo the maladaptive process, and this entire process, the angiogenesis of fibrosis will go up, and collagen production will go up, and you will see cell death and apoptosis, necrosis, and so depending on the type of stimuli, then the myocardial death will occur and leads to heart failure. So in cardiac field, heart failure is not considered as a disease, but hypertrophy is considered as a disease. So that's why, the early diagnosis, hypertension or anything, people will try to prevent the hypertension or diabetic conditions to prevent the heart to undergo, pathological hypertrophy and progress to heart failure. So we're going to see one example, which is the last part of this lecture, which is a cardiac ischemia. So ischemic heart disease is one of the most prevalent heart disease in the world, and it is caused by imbalance between the myocardial blood flow and the metabolic demand. So you will see a reduced blood flow, but increased metabolic demand. So there is a mismatch between the oxygen supply and the energy or ATP production. And the myocardial ischemia is mostly caused by the reduction in coronary blood flow, and it is mainly related to atherosclerosis. We'll see in the next slide what is atherosclerosis is. So in the myocardial ischemia could be two types depending on the obstruction of the coronary arteries. If it is partially occluded, then it's called angina. There will be insufficient amount of oxygen supply. Therefore, the rest of the myocardium will work hard to produce more ATP molecule, ended up with hypertrophy and heart failure. And then the second type of ischemia is myocardial infarction, which is a total occlusion. So if the coronary artery is completely occluded, and then there won't be any blood flow or oxygen supply to the wherever it is occluded, and that will cause us the myocardial damage and cell death, and it will be replaced by fibroblast and scar tissues. That's called myocardial infarction. So atherosclerosis is one of the the leading cause of the cardiac disease, myocardial ischemia. This is mainly by the accumulation of cholesterol-rich lipids in the intima of the large arteries and coronary arteries. So it can occur in other arteries also. We're going to see it in the next slide. But if it occurs in coronary artery, it is called as coronary artery disease. So this the atheroma is nothing but the lipid pool, which will be covered by fibrous cap. And this fibrous cap will kind of started to grow depending on the accumulation of the lipid, and then thereby it will reduces the lumen of the artery and blood supply. Okay. And this could be like this. So this is a normal blood flow. And if there is an atheroma, it's going to grow inside, and thereby it will kind of prevent or it will restrict the blood flow. Or if it is ruptured, then it's going to completely block the blood flow. The atherosclerosis, it's a very slow and progressive buildup. It contains fatty substances, cholesterol, cellular waste, calcium, and fibrin. If it is not treated, it's going to suddenly block the portion of the heart, depending on the type of atherosclerosis and the severity of the atherosclerosis. So this is one of the major causes of myocardial ischemia. So the other type of atherosclerosis are the carotid artery disease, which is the plaque builds up in the carotid arteries, which supply blood to the brain, and that leads to stroke. And peripheral arterial disease, here the arteries, the plaque forms in the arteries which supply blood to the leg, arms, and pelvis. Um, and the third one is abdominal angina, bowel infarction. This is the narrowing of the artery supplying blood to the intestine. This causes the abdominal pain and abdominal angina. So all sort of, the atherosclerosis can feedbackly affect the cardiac function in a different extent to a different degree. So, in some cases, like, even the coronary artery disease can contribute to some of the peripheral artery diseases. So if there is a blockade in the coronary artery, the cardiac function will be weak and then the pumping efficiency will be reduced. That will also causes the peripheral artery diseases. So it's kind of like, in both ways, and then it contributes to hypertrophy and heart failure. So other than the atherosclerosis, there are other factors which can also reduce the coronary blood flow. So the first one is decreased aortic diastolic pressure. So the aorta also contracts during the cardiac cycle. Once the ventricle contracts, the blood will be pumped into the aorta. Then the aorta will contract to pump the blood for the systemic circulation. And if this pressure is reduced during the diastole of the aorta, during the aortic relaxation, and then it will reduces the blood flow of the coronary arteries, because coronary artery is the first branch of aorta. So if there is a defect in the aortic contraction and relaxation, that will affect the coronary artery. And second one is the increase in intraventricular pressure and myocardial contraction. The ventricle develops a certain amount of pressure. It's around like 140 millimeter mercury during contraction. If the pressure reduces, the pumping efficiency will reduce. That will also reduces the flow of the blood to the coronary arteries. So that contributes to contribute to myocardial ischemia. Other than that, coronary artery stenosis, again, narrowing of the coronary artery, and vasoconstriction, aortic valve stenosis, regurgitation, and increased right atrial pressure. So all these factors can contribute to myocardial ischemia. So what is the functional consequences? Myocardial ischemia induces both systolic and diastolic dysfunction. So systolic means contractile dysfunction, diastolic means like relaxation defect. So systolic dysfunction is characterized by impaired contraction. This is mainly due to the lack of oxygen and accumulation of end products, like, the metabolic activity will go down. So you will see the accumulation of inorganic phosphates, protons, lactate, and also the potassium ion transport will be reduced. So all these will reduces the membrane depolarization and contractile ability. Then the second one is the diastolic dysfunction. This is referred to the impaired relaxation. This is mainly due to the lack of ATP. As I said before, the three molecules, myosin, SERCA, and sodium potassium ATPase, these are all the three molecules which are utilizing most of the ATP synthesized by the cell. The myosin ATPase utilizes 50%, SERCA utilizes like 25%, and sodium potassium ATPase utilizes 15% of the ATP synthesized by the cardiac myocyte. So if there is no oxygen supplied to the myocardium, the oxidative phosphorylation will go down. So that will result in reduced amount of ATP synthesis. And since the ATP level goes down, it will affect all three molecules, which are important for muscle relaxation. So if there is no ATP, and the myosin will be in a contractile state, it's not going to relax. I mean, if you remember the actomyosin interaction, the ATP binding is important for the muscle relaxation. So without ATP binding to the myosin head, the myosin cannot detach from the actin filaments. So it will be in a contractile state. SERCA is important for the calcium restore into the SR, and without SERCA activity, the calcium will stay in the cytoplasm, so there won't be enough calcium available in the SR in a long run for the contraction, and also the muscle is not going to relax. Then the sodium potassium pump activity is also reduced, so that will result in the accumulation of cytosolic sodium. The pump cannot take the sodium out. So if the sodium is more inside, the cell will be in a depolarized state. It's not going to repolarize. So that's going to impact the muscle relaxation. So in a ischemic condition, therefore, you will see a systolic dysfunction and diastolic dysfunction. And so if there is no oxygen supply, as I said before, the myocytes are going to die, and it will be replaced by the connective tissues. And then the fibroblasts will produce more collagen molecule. And initially, it will be like a compensatory process. But however, if it is not treated, then it's going to impair the function. So this one, because of this, you are going to see the entire LV is dilated and remodeling, which is a hypertrophic process. And then finally, you will end up with the cell death, which is infarct, and then more dilated ventricle with a less amount of cardiac muscle. So that will impair the muscle contraction and relaxation. So this is, results in heart failure. So the symptoms of heart failure or cardiac ischemia are, first is pain, which is due to the accumulation of adenosine. Because since there is no ATP synthesis, and the system will try to use, make the ATP from ADP molecule and AMP, finally you'll end up with the adenosine. It will be finally broken into ATP, into ADP, and then AMP, then to adenosine. And then the adenosine accumulation can stimulate the nerve endings, which will cause us the pain. And then syncope, which is a transient loss of consciousness. And this is due to the atrial arrhythmias, both ventricular and atrial arrhythmias, and vagal stimulation and massive myocardial dysfunction. Arrhythmia is the random contraction, of the cardiac muscle. Nausea, this is due to the irritation of the diaphragm and by the inflammatory reaction. Then irregular heartbeats, this is a sign of arrhythmias created by the ischemia of the conduction system. And finally, dyspnea, which is a difficulty in breathing. This is due to the accumulation of blood in the right circulation. Since the cardiac muscle, the left ventricular function is reduced, most of the blood will be in the right ventricular side. It started to accumulate in the vein and venous system. So that will cause us the pulmonary edema, because the most of the blood will goes to the lungs, and it will not be pumped properly. And then with the pressure, it will cause the pulmonary edema. So, and this causes the difficulty in breathing. I think that's it. With this, I will stop here. It's very difficult to combine everything in a cardiac muscle, because I have a course called Molecular Medicine of the Heart. So we teach the whole thing in detail, and I kind of comprised everything into small portion. So for the exam point of view, just go with a concept and important, the structure and the disease related with it. So that will help you. You'll also get a sample questions from me. So any questions? Okay. If no questions, I'll stop here. Welcome.